So hello, welcome to the Yorkshire Philosophical Society for the first of three linked events we're hosting this year on antimicrobial resistance. And it gives me great pleasure this evening to introduce our first speaker in this series, Professor Jessica Blair from the University of Birmingham. I'll tell you about her in a second. So I don't know if you've seen our flyers. We have these three linked talks, the first one this evening, another talk in June, and another talk in October, which Jess might mention in her talk. So these are three linked sessions from three prestigious UK academics on this theme of antimicrobial resistance. So hopefully by the end of the year you'll have a really good picture as to what antibiotic resistance is and where there's potentially new hopes in finding ways to combat it and get new antimicrobials. Right, so our speaker this evening is a professor of microbiology at the University of Birmingham. Jessica did her degree in Oxford in biological sciences and then moved to Birmingham to do a PhD with Professor Laura Piddock, who also is a very well-known professor who works on antimicrobial resistance in the UK and has been very important in some of our government policy and educating people about AMR. Jess decided that was a great place to do a postdoc, which she did in the same laboratory in the medical school at the University of Birmingham. And then she won a very prestigious BBSLC David Phillips Fellowship to enable her to start her own career in Birmingham and start her own group. And she's never looked back, so she's been there since and got promoted to a personal chair in microbiology in 2023. And I've known Jess for quite a long time. She was an editor with me when I was editing Microbiology, which is a UK learned society journal. In fact, we created a new section on AMR, and Jess came in as one of our key editors. But soon afterwards, she was poached by a Nature journal who were founding a new journal on antimicrobials and resistance, and Jess is now editor-in-chief of that journal and really looks after a lot of really important research in this area globally, as well as her research in the UK. So this evening she's going to kick off our three talks. She's going to tell us about what is a superbug and introduce us to the global threat of antibiotic resistance. So can we thank Jess for coming up from Birmingham this evening.
Good evening, everybody. Thank you so much for the invitation to come. It's really, really wonderful to be with you tonight. So I'm going to tell you a little bit about superbugs, a little bit about antibiotic resistance, and we're going to look at some of the words associated with this. We're going to try and figure out what a superbug is. We'll come back to that question later and try and answer it. So first of all, we're going to talk about some words that you will almost certainly think you know what they are. This isn't advancing. Okay, we'll do it this way. Okay, so we're going to look at some words that you will almost certainly understand, know what they mean, but we're going to think about what the actual definitions are before we start. So, an infection. This is a word that you will all have used many times, particularly over the last 5 years or so. But what does that actually mean? Well, an infection is really the invasion of an organism's body tissues by pathogens, disease-causing agents, such as bacteria. So we've got bacteria in green at the top over there. We've got viruses in blue. We've got fungal infections here. So fungal infections could be things like athlete's foot or thrush. Or we've got, if you want to give yourself nightmares, Google parasitic infections. This is a tapeworm here. We're not going to talk about many of those today. What we're going to focus on are these guys, bacterial infections. So again, let's think about what that means. So what are bacteria? Well, they're really small, microscopic, single-celled organisms. And they come in all shapes and sizes, round ones, rod-shaped ones, spiral ones. And really, when we look at them down a microscope, they might look something like this. And when we grow them in the laboratory, we do that on agar plates like these. This is a jelly-like substance that we grow the bacteria on, and they look something like this in all different colours. So they're very small, single-celled organisms. And these can grow and divide very quickly. So bacteria grow and divide by literally one cell divides into two. So what that means is, Let's try that again. What that means is that they can replicate really quickly, so we can quickly go from having 1 bacteria to 2 to 4 to 8 to 16 to 32 to 64 to 128 to 256, and suddenly we've got a phenomenal number of bacteria. And actually, bacteria are everywhere. So every single one of us has 100 trillion bacteria associated with our bodies, so inside and on our bodies. Now, 100 trillion is a kind of crazy number, but a bit difficult to understand what that really means. So if we think about, if I gave you a dollar bill for every bacteria that's in or on your body, and we stacked them on top of each other, they'd reach to the moon and back 14 times. If I spent $20 a second, it'd take me 158,000 years to spend the money. So this number is phenomenal, right? Absolutely enormous number of bacteria that lives with every single one of us. And they're not all bad. We're talking about infections, but most of those do us no harm whatsoever, and many of them are really important to keep us healthy. But we're talking about antibiotics today and infections. When we think about infections, we might think about feeling unwell, about going to the doctor, about being given an antibiotic. So what does an antibiotic, what does that actually mean? Well, the translation, I suppose, is literally sort of the anti is reversed sort of against, and the biotic is life. Antibiotics are powerful medicines, and they fight bacterial infection. The reason I've put bacterial in bold text here is because antibiotics only treat bacterial infection, okay? Taking antibiotics is not going to help you with the common cold, which is caused by a virus, or with the flu. It's not going to help you with a fungal infection. They only treat bacterial infections, and that's a really important point from tonight's talk. So, in 1900, Paul Ehrlich was a scientist that coined the phrase magic bullets when he was talking about antibiotics. And what he meant by that was antibiotics being something that would kill bacterial cells, but not kill the human cells that surrounded them. Killing bacteria is easy, right? Bleach kills bacterial cells, but I can't give you that to treat your sore throat or your infected toe because it'll do more damage than the infection itself. So the idea of Paul Ehrlich introduced was that we were looking for molecules that could kill bacteria, kill infections, but not kill us. And actually, he developed the first antibiotic in 1909. This was a drug called salvarsan that was used to treat syphilis. It's not a great antimicrobial, to be honest, and it's very toxic. But perhaps more famously, Alexander Fleming discovered penicillin in 1928. Now, some of you might be aware of the story of how Alexander Fleming discovered penicillin. Essentially, he was a microbiologist, and he went away on holiday, and he had a messy laboratory, perhaps a scientist after my own heart. And he had a stack of these agar plates that I spoke about. And you can see that you've got bacterial growth on... These don't work. You've got bacterial growth across the plate in the sort of zigzag bit. That's the bacteria that he was studying. And when he came back from his holiday, he'd left the window open and he could see that he'd got this blob of mould growing on his agar plate. That's not unusual. We see contaminants growing on agar plates a lot of the time. But what he noticed was that, can you see around the blob of the mould at the top, there's a zone where the bacteria have not been able to grow. And he looked at this and thought, well, this is interesting. What is it stopping the bacteria being able to grow in that zone? Many years later, we know that that is penicillin, the antibiotic that many of us may have taken. And in 1945, he won the Nobel Prize for Medicine for the discovery of penicillin, but along with these two guys here. So Ernst Chain and Howard Florey were really, to be honest, the brains behind the operation in this story, because while Fleming discovered penicillin, these were the two scientists who figured out how to isolate it, how to get enough of it to make it into a useful antibiotic. So the three of them shared the Nobel Prize for Medicine in 1945. And really, the discovery of penicillin was a discovery that changed the world. There's lots of studies looking at how treatment with antibiotics have changed world history, but one example is the Second World War. So penicillin was introduced in the 1940s and was used to treat many soldiers during the Second World War. 2.3 million doses of penicillin were produced in time for the Normandy landings in 1944. And what that meant was that thousands of soldiers that would previously have died from infections and from sepsis survived to either continue fighting or survive to be able to come home. So that's sort of a historical view of penicillin, but what about if we bring that back to today? Can I have a show of hands in the room of anybody, who in this room has taken an antibiotic in their life? Pretty much all of us. And actually, let's think about that for a minute. We've all taken antibiotics to treat infections that we've had. Now, some of you would have got better anyway. It might have taken longer, but you might have got there. Some of you might not. Some of you have probably had your lives already saved by the fact that you were able to take an antibiotic to treat that infection. Now, we know that life expectancy in the UK has increased significantly over the last 100 or so years. So in 1900, the life expectancy in the UK were 47 and 50 for men and women. And now those figures sit at 79 and 83. So it's an enormous difference. There's lots of things that have caused that. We've had better sanitation, clean water, we have better healthcare in all sorts of ways. But estimates suggest that 23 years of that difference is because we're able to treat infections with antimicrobials. Before we had antibiotics, infectious disease killed 20 to 30% of children before the age of 10. Oh, sorry, and I suppose, and then the final part of this is that now 1 in 4 babies born today can expect to live to the age of 100. So antibiotics, pretty important stuff. And actually, antibiotics underpin all of modern medicine. All of the medical procedures and treatments that you see here would not be possible without antibiotics. Each year in England alone, about 4 million operations take place. And here we use antibiotics both pre- and post-operatively to prevent infection. 1 in 4 births in the UK is now by caesarean section, and here we use antibiotics to protect both mother and baby. Half of women in the UK will experience a urinary tract infection within their lifetime, and many of those will require antibiotic treatment. Most cancer patients, or most cancer treatments, suppress the body's immune system, making it difficult for cancer patients to fight infection. So here, antibiotics and antifungals and antivirals are absolutely critical to keep people alive while they take their life-saving cancer treatments. Meanwhile, anybody who's had a transplant will know that their life depends on antibiotics to treat life-threatening infections. So here's a question for you. Without effective antibiotics, would you opt for routine surgery, something like a hip replacement, if the risk of death was unacceptably high? Or would you soldier on with your dodgy hip? Difficult questions to answer. But really the point of this is that we rely on these magic bullets, and without them, our healthcare system would look completely different. So after the discovery of penicillin, there was really a golden age of antimicrobial discovery. And discovery of antibiotics took off at pace between the sort of 30s and 60s, when lots of different classes of antibiotics were discovered. And previously fatal infections were now easily curable with our magic bullet drugs. Now, scientists and doctors, particularly doctors, like to think they're really clever, right? And around the early to mid-1960s, there was a general feeling among the scientific community that, hey, we've sorted this. Infectious disease is kind of done. And there's lots of quotes like this, but this is one from Sir Frank McFarlane Burnet, who was a Nobel Prize winner. His work was around the immune system. And he said, "One can think of the middle of the 20th century as the end of one of the most important revolutions in history, the virtual elimination of infectious diseases as a significant factor in life." Well, anyone who's lived through the last 5 years or so can attest to the fact that that isn't true, and he got that wrong, sadly. Now, there's many reasons that hasn't turned out to be the case, but one of them, and the reason that we're all here tonight, is to talk about this. And that is antibiotic resistance. So this is the fact that many of our antibiotics don't work that well anymore. So what antibiotic resistance means is that bacteria change in a way that makes them able to survive treatment by antibiotics. So if there's, a really common misconception when we talk about antibiotic resistance is that people become resistant to antibiotics. That's not the case. What happens is it's bacteria that become resistant and we're no longer able to treat them. What also happens is that bacteria can become resistant to multiple antibiotics at the same time. So that's what we call multi-drug resistant bacteria, and that is pretty scary stuff. But what does this actually mean? This means that you could get an infection that can't be treated by antibiotics. Now, hopefully, your immune system would cope with that and you would get better. But if it doesn't, you might die. No major surgery would be possible, no organ transplantation, no cancer treatment. Antibiotic resistance is pretty serious stuff. This timeline is an interesting one. So at the top of the timeline, you've got when different classes of antibiotics were introduced into the clinic. Okay? On the bottom, you've got when we found bacteria that were resistant to that drug. So the first thing to notice is that every drug that appears on the top also appears on the bottom. So we've got resistance to every antibiotic that's currently licensed to be used in the clinic. Okay? Not necessarily hugely widespread, but we've found resistance to all of these drugs. The amount of time between when they were introduced and when resistance was found varies, and the rates at which resistance is prevalent varies, but the fact that there is resistance to every single drug is a scary thought. So perhaps now we can start to think about the question that I posed at the beginning in my title. So what is a superbug? Well, the first thing to say is you've probably all heard this phrase because the press love a superbug story and we see them in the news, lots and lots, hospital superbugs, and we see lots of things about this. And actually, I don't think, sometimes the press don't get that right, they don't use it appropriately, but I thought it would be useful to sort of pin that down and understand what that actually means. So what is a superbug? So superbugs are pathogens. So what that means is those things we looked at on that first slide, bacteria, fungi, viruses, and parasites as well, which are resistant to multiple antimicrobials. So we're talking about bacterial pathogens today. So we're talking about bacteria which cause infection that are resistant to multiple antimicrobials. That is what a superbug is. So I thought it might be useful to talk a little bit about how antibiotics work. So we're saying they're these wonder drugs, aren't they amazing? But actually, what do they do? How do they work? We can look at this in two slightly different ways. So on the left, when we've got no antibiotics, bacteria are able to multiply. They'll continue to grow and divide. In the middle, we've got the idea that we can have bacteriostatic antibiotics. Now, what that means is some drugs don't necessarily kill the bacteria, but they stop them being able to divide. So we don't get the situation like I showed in that video. Or, on the right-hand side, we have some antibiotics which act as bactericidal agents, which means they actually kill the bacteria that are causing infections. So there's two different ways that antibiotics work. And we can have a look at another video, actually, that shows us this. So this is penicillin. This is a bactericidal antibiotic. What you're going to see on the left-hand side is these are bacteria with no antibiotic, so they're going to start to grow. And on the right-hand side, we've got what happens with this antibiotic penicillin, which is killing the cells. You'll start to see is they start to explode on the right-hand side. So we're literally killing those bugs by adding this antibiotic. So really what antibiotics do, and don't know too much about the details here, is they target parts of the bacterial cell that are absolutely required for life. So if you target something that's required for the bacteria to live, then the idea is that you'll be able to kill it or stop it from being able to divide. So really I've sort of split these into four different categories. So we've got, in the top left, cell wall inhibiting antibiotics. So the cell wall is the outside part of the bacterial cell that's really important for keeping the bacterial structure, I guess. And lots of antibiotics target this. We also have some that target a sort of membrane which sits under that cell wall. And at the bottom we've got two different categories here. We've got antibiotics that target nucleic acid synthesis. So this is target the ability of the cell to make DNA or RNA. So if it can't do that, it can't grow and divide. Or there's there's antibiotics that target the cell's ability to make proteins. Proteins are the building blocks of life. If they can't do that, then the bacteria will be killed. So there's lots of different ways that antibiotics work, but we need to have some understanding of that to understand how bacteria become resistant to them. So how do they stop being killed by antibiotics? Well, bacteria can do really two things. They can develop mutations within existing genes in their chromosome that mean that antibiotics don't work anymore. So that's genes that already exist that just change a little bit. But bacteria are also able to acquire completely new genes and share them between each other. So genes which code for the ability to not be killed by a particular drug can be shared around between different bacteria, and this is how antibiotic resistance can spread very quickly. So let's do a little quick demo about how antibiotic resistance spreads and how antibiotics stop working. So I'm going to need everybody to take part in this. So I want everybody in the room to pretend that you are a bacterial cell. You're all bacteria causing an infection. Let's say you're causing me to have a really sore throat, okay? When we think about human populations and the people in this room, we know that humans vary. And we think about those variations as being eye colour, hair colour, skin colour, shoe size, right? And those things are really obvious. With a bacterial population, there's still variation. It might not be as obvious. It could be a very slight change in one gene within the whole bacterial chromosome that makes very little difference really to how the bacteria lives most of the time. Right, so live bacteria all have their hand up. So if everybody is able, can put their hand up as you are a live bacteria. Okay, great. So I've got a sore throat. I've got a bacterial infection in my throat, and I go to the doctors, right? And the doctor gives me an antibiotic. Great. I take the antibiotic, and what we hope happens is that all of the bacteria in the infection are killed, so everybody puts their hand down. Great. In this scenario, I feel better, and I go back to work, I get on with my life, and forget all about it. But actually, let's think about a different scenario. So let's have hands up again. So in this scenario, I've got a sore throat, I go to the doctor, I get given an antibiotic, but this time there just happens to be some variation in the bacteria that are causing the infection, and there's certain bacteria which just aren't killed by that antibiotic. So in this case, the antibiotic kills almost all the bacteria, but bacteria wearing a checked shirt don't get killed by the antibiotic. So here, most of the bacteria die. Right, I start to feel a lot better, okay, and perhaps my throat feels better, I might go back to work. But now, I haven't actually killed all the bacteria that are causing the infection because there's a small number of them left. Now, if we think about that video where I showed cells dividing, these bacterial cells are going to start to grow and divide. But now I've got an infection in the back of my throat, I might start to feel unwell again, but it's caused by check shirt bacteria, right? So thank you very much, you can put you down. So you get the idea, right? So there was just a few of them, but they're going to start to grow and divide. Now the infection in my throat is caused by check shirt bacteria. I might go back to the doctor and get given some more of the antibiotic. But now it doesn't work because we know that check shirt bacteria aren't killed by the antibiotic that I've been given. What we've done is we've killed off all the susceptible bacteria and we've just selected for those that are resistant to the antibiotic, and they're the ones that have survived and go on to cause the infection. If I now pass that infection on to my husband or my son or someone that I work with, they've now got an infection with check shirt bacteria and antibiotics will be less effective. So we can see this in the same way on this slide. So we've got a population of bacterial cells, we take an antibiotic, most of them die. A few of them survive because they happen to be resistant to that antibiotic. They grow and divide, and now the antibiotics don't work. So what doesn't kill them makes them stronger. So actually, this video, I think, is absolutely terrifying. So this is an experiment that was done at Harvard Medical School in 2016 by Michael Bowman and the team he was working in. And what you've got here is a really big agar plate. This was called the mega plate, and it was massive. And you can see there's different zones. So in the different zones, you've got different amount of antibiotics. So you're going to see in a minute that bacteria are inoculated at the edge of the plate where there's only a very small amount of antibiotic. And in each section, there's 10 times more antibiotic than there was in the section before. So what you see is that bacteria starts to grow at the edges where there's not much drug. Then you'll see that we get a breakthrough of cells being able to grow in a higher concentration of drug, and within days they're able to grow now at 100 times the original concentration of antibiotic. Now we're selecting for further resistance, and in a minute we'll get to the point where they're able to grow in the middle section where there's 1,000 times more antibiotic than we started with. So in a matter of days, bacteria with this selection for antibiotic resistance has really meant that they can grow at such high concentrations of drug. So how do they do it? This is really what my research is about different mechanisms by which bacteria become resistant to drugs. And I'll just briefly tell you about some of the ways they do it. First of all, bacteria can acquire the ability to destroy antibiotics, so they can make enzymes that actually chop up the antibiotic molecules and they don't work anymore. Or they can dodge the antibiotic. So ways they can do that, they can have an alteration in the target of the drugs. This is a bit like our check shirt bacteria. We get a mutation that means that the antibiotic doesn't work anymore. Or they can find a different way to do a process that is absolutely required for life. So if an antibiotic is inhibiting one way that a process happens, they find a different way to make that process happen. Now what I'm going to talk a bit more about is the next two. So a lot of antibiotics work by getting inside bacterial cells, where they bind to a particular target and they either stop the bacteria being able to divide or they kill the cell. But they need to get inside to do that. And the more drug that gets inside the cell, the more likely we are to be able to kill that bacterial cell. But one thing bacteria do is they try to prevent drug getting in. They make it really difficult for antibiotics to enter the cell. And also, they can pump the antibiotic molecules straight back out of the cell again. And this is through something called efflux pumps that we're going to talk a little bit more about. So, efflux pumps are found in every living cell. So whether that's an animal cell, a plant cell, a bacterial cell, they will have little molecular machines on the surface of the cells that pump stuff from inside cells to outside cells. And they pump all kinds of things and are really important for life. But when we're talking about bacteria and we're talking about antibiotics, the reason these pumps are important is because they can pump antibiotic molecules from inside bacterial cells to outside. And that means that the concentration inside the cells remains lower, meaning that bacteria can survive at higher concentrations of drug. Now, these machines are actually quite complicated on the right-hand side, but I'm going to show you a little bit. Well, so what happens is, so these molecular machines pump antibiotics out. These look quite complicated, but I'm going to show you this in a much simpler way. So I've got a bit of a demo. Right. So, okay, can everybody hear me through this mic now? Yeah, good. Okay, this is a bacterial cell. Okay, I'm going to need a volunteer. Well, a volunteer, Gavin. Right, you can take that. Okay, so we've got our bacterial cell and we want to be able to kill this bacterial cell. And I said that the more drug we can get into the cell, the more likely we are to be able to kill it. So if we say to kill the cell, we've got to get antibiotic up to this blue line. Okay. Gavin has got a watering can with antibiotic in it, okay? And he's going to pour it into this bacterial cell and see if he can kill the cell. Go for it. So quite simple, right? He's pouring the water in. We're pretty soon going to reach the line, and we're going to say that that bacterial cell is dead, hopefully. Maybe we might need to draw the line lower. We're getting there. So you can see that this was quite straightforward. Okay, you can stop. You can pour that into there now. That's okay. Right, I'm going to need another volunteer, if that's okay. Someone who wouldn't mind pumping this? Yes, perfect, thank you. Okay, so now I'm going to show you why efflux is important. time, we're going to introduce an efflux pump. Okay, so I just told you that these are pumps that pump antibiotics out of cells. I'm going to give this to you. Right, so this time, Gavin's going to pour the antibiotic in to the bacterial cell, and we're going to try and pump it out. I'm going to try and hold that over the bucket. Okay, so let's go. Yeah, pump it in. So if you keep pumping, hopefully. Oh, there we go. What we can see now is that water is being pumped out by the efflux pump. It's now quite difficult for Gavin to kill the bacterial cell because as fast as he pours the water in, the efflux pump is pumping the water out. Okay, perfect. We're going to do one more. Okay, we'll reset. Thank you. We'll do one more. Okay, you done with me? No. Okay. Oops. Right. So this time, I'm going to show you what we're trying to do in the research that I'm doing. What I've done is I've made this pump not work so well. I've added a cable tie, okay? So now we're not going to be able to pump it. So whereas last time the efflux pump pumped the water out really well, and Gavin was finding it really difficult to kill the bacterial cell. That's last time. Now the pump doesn't work. So perfect, thank you. I think we get the idea, that's great, thank you. So, put that down, thank you. Right, so in this analogy then, the cable tie is what we call an efflux pump inhibitor. So what we're trying to do in my research is figure out how these efflux pumps work because they're these pumps that pump antibiotics out of bacterial cells. And we're trying to find ways to stop them working. So we're trying to find molecular cable ties. We used a cable tie here. We're trying to look for drugs that would do the same thing. So we could give a drug with an antibiotic and stop the efflux pumps from working and make bacteria susceptible to drugs again. Thank you very much for your help. So this slide sort of shows exactly that again. So on the left-hand side, we've got the situation where we've got efflux pumps working. They're pumping antibiotics out of bacterial cells, so the concentration inside the cell remains low, and these cells are resistant. On the right-hand side, we've got a situation where the efflux pumps aren't working. This is my cable tie, and antibiotics then accumulate much better within the cells, and the antibiotics work much better. Okay, so we've talked a lot about antibiotic resistance. We've talked about what it is, how it works. But really, I wonder whether the reason you're here is this second bit. How bad is it and why should I care? So I'm going to show you some stats and some numbers now that really illustrate how bad this is. So current estimates are that there are around 5 million deaths associated with antibiotic-resistant infections every single year, of which about 1.27 million are directly attributable to AMR. So if we think about that 5 million deaths, that's 1 every 6 seconds. So today, on the 20th of May 2025, at 7 o'clock, so at the beginning of this lecture, so far this year, 2,019,333 people had died and their deaths were associated with antibiotic resistance. By the end of this lecture, there'll be another 600. And perhaps even worse than that is that 1 in 5 of those deaths occur in children under 5. And as with many things, the rates of AMR are not equal across the globe. And actually, low and middle income countries are far more affected than other parts of the world. And you can see here, so this is mortality rate essentially across the world. And you can see that the worst affected area on the globe is sub-Saharan Africa. So what about the future? So this was a report that was commissioned nearly 10 years ago now. And experts debate the exact numbers, but the principle remains that if we don't do anything about AMR, estimates suggest that by 2050, 10 million people a year will die because of antibiotic-resistant infections. That's more than cancer and diabetes put together. It's not just a health problem, it's also an economic one. And this would cost the global economy £66 trillion. And that would plunge an extra 24 million people into extreme poverty. So it's both a health and an economic issue. So why is it so bad? Well, let's look at a few different reasons. Let's talk about, first of all, antibiotic use in humans. So greater antibiotic consumption, we're using more and more antibiotics, and I'll show you a graph in a moment. And some of the time, that's absolutely the right thing to do, and we should be able to use our antibiotics to treat infection. But sometimes they're prescribed when they're not needed, or for an illness that isn't going to be made better by antibiotics. So, for example, we know that only 10% of sore throats benefit from antibiotic treatment because they tend to be caused by a virus. Yet, antibiotics are prescribed in as many as 60% of cases. A recent study found that nearly a third of antibiotics prescribed by doctors in the US were prescribed for conditions that don't respond to antibiotics. So that equates to 47 million unnecessary prescriptions a year. So that's driving this antibiotic resistance crisis when it's not needed. And this is just a graph to show you that globally we're using more and more of these drugs. It's important to say, though, that the pattern is different in different countries. In England, total usage has fallen. But if you think about the UK, 1/3 of us will take a course of antibiotics in any given year. So we're using a lot of them. So why else is AMR so bad? Well, we also think about how we use these really important, powerful medicines not in human usage, so things other than treating humans. So the first thing is we use them a lot in animals. So we use them to treat sick animals. These are my dogs. The one on the right-hand side is an elderly Labrador called Dexter, who recently had some doggy coamoxiclav and it saved his life. So I'm not for a moment saying that we shouldn't be doing that. But we use them a lot in animals. We use them a lot in agriculture. And in some cases, when they're used in agriculture, they're given to a whole herd or a whole flock, rather than necessarily just one animal. And globally, around 75% of all antibiotics are actually used in agriculture. And sometimes this is in something called growth promotion, where animals will be given an antibiotic in their feed because it helps them to grow bigger and stronger faster, so that the farmer can make more money. So it's not a simple one. There's good reasons that has happened, but it's certainly something that is driving the AMR crisis. We also use biocides a lot. So biocides are things that you will find in household products like these, which you've almost certainly got in your house. Things like antibacterial soap, toothpaste, shower gel. If any of them say antibacterial on them, they almost certainly contain a biocide of some kind. Microban is a big trade name that you might have. They make chopping boards and fridges and things that say Microban on them because it's supposed to kill bacteria on those surfaces. This is an antibacterial stapler. I'm sorry, nobody in the world needs an antimicrobial stapler. These contain a compound called triclosan. The problem with using lots of these compounds where we don't need them is that they also select for antibiotic-resistant bacteria. They're also used a lot in aquaculture. So if you're a salmon farmer and some of your salmon have an infection, you don't catch each salmon and give them a dose of drug, you put it in the water. So we're using a vast amount of antibiotic and essentially releasing it into the environment, etc., etc., etc. So there's lots and lots of uses that are not human use of antibiotics. And if we look at antibiotic consumption by country, it varies hugely across the world. And is set to, this is a very recent study suggesting that antibiotic usage is actually set to increase if we carry on as we are. The other reason that AMR could be such a big problem is because we don't have many new antibiotics. I said earlier that there was this golden age of drug discovery where new drugs seem to be discovered all the time, but that is not the case anymore. So we had this golden age of antimicrobial discovery, followed really by this discovery void, which found all the very easy, low-hanging fruit antimicrobials there were to find, and resistance problem was growing. So it was much more difficult to find antibiotics. So really, we absolutely need novel therapeutics to combat antibiotic-resistant infections. And this is where I want to give a plug to the 2 other talks that are coming up. So the next one is Professor Martha Clokey, who's going to talk to you about bacteriophage. These are viruses that kill bacteria. So it's an alternative to antibiotics. It's super cool. You should definitely come and listen to Martha talk about that. And the final talk in the 3 is Kate Duncan from Newcastle University, and her work is looking for new antibiotics, and she goes to really cool places, Antarctica, the deep sea, looking for really cool molecules that can kill bacterial cells, so you should absolutely come to both of those talks too. The problem is, it's really hard to find new antibiotics, and it's really Martha and Kate that are going to tell you more about this, but I'll just give you a bit of an overview of why this is the case. So here's some scientific reasons it's difficult to make new antibiotics. An antibiotic has got to be a magic bullet, right? It's got to be selective. It's got to kill bacteria without killing you. And that's really quite a challenge. It's got to last long enough in the body, and it's got to get to the site of infection. So if you take it as a tablet, it's got to get to the part of your body that's infected. So this is what we call pharmacokinetics and pharmacodynamics, making sure that the drugs work within the body. This is really important. There's no point making a new antibiotic that works brilliantly in the lab if actually there's already loads of widespread resistance to it. If bacteria already know how to not be killed by that drug, well, there's no point, right? So you've got to find a drug that there's no existing resistance to, and really the way that you do that is you find drugs that work in a completely new way. So you're looking for new chemistry, new mechanisms, and that's really scientifically challenging. The other reason it's hard to develop new antibiotics is economics. They're really expensive to develop, and they take a long time. Lots of drugs fail along the pathway, which means for everyone that gets through, it really is a hugely expensive process. It's really difficult to get them licensed, to be able to give them to people. And that means that, all of this means that the potential revenue is lower. You know, if I was a drug company and I had the choice between making something like a statin that somebody might take every day for the rest of their life, or I could make an antimicrobial that somebody might take 3 times a day for 5 days, get better and never take again, you can see which one could make the most money. Then the other thing is, if you make this amazing new magic bullet antibiotic that works brilliantly, people like me are going to say, don't give that one out too much because we'll get resistance. Let's keep that for when we really need it. So actually, it can be quite difficult to make money from antibiotics. So lots of the big pharma companies have pulled out of making them. There are still some, and there's lots of work going on to figure out how the economics could be changed and different models for how money could be made on antibiotics so that these companies stay in that space. Really, I suppose this could be the killer slide in the whole thing. This is what happens if we don't crack the problem. So there's 2 quotes here. On the left-hand side, we've got a quote from Margaret Chan, who was the Director General of the World Health Organization. And she said that a post-antibiotic era, in effect, means an end to modern medicine as we know it. Things as common as strep throat or a child's scratched knee could once again kill. And if we look on the right-hand side, Dame Sally Davis, who's the UK Special Envoy for AMR, says that without new antibiotics, we will move into an apocalyptic phase. And that's all pretty scary stuff, right? That's what we're talking about. We're talking about not being able to treat infections and not being able to have healthcare as we currently know it. But I didn't want to end on doom and gloom. Let's end on some good things and what's happening to sort this out. So what's happening globally? The really good news is that world leaders are listening. There was a big agreement at the end of last year, signed up to by lots of member states and committed to a clear set of targets. So they've signed up to reduce that number of deaths by 10% by the year 2030. Doesn't sound like a lot when the numbers are huge, but it's hopefully a start. And it's also looking at dealing with the problem and providing basic water sanitation in healthcare facilities, and making sure that at least 60% of countries have national antimicrobial resistance plans. But what else needs to happen? Well, we need to use better stewardship. So this is using the drugs we already have well, making sure that we use them in a way where we don't add to the resistance problem. We need rapid diagnostics, so we need to be able to, at a patient's bedside, decide whether they have a bacterial infection. Are antibiotics going to work for this patient? And is the bacteria susceptible to the antibiotic we want to give? This process takes a couple of days. We need to be able to do that much quicker. We need improved surveillance to know what resistance is happening, and we need new drug discovery, as I say. Martha and Kate are going to talk to you all about that. So just a final note then. What can we all do? It's all very well thinking about what the global leaders are doing, but actually what could we do? Well, the first thing I would say, the absolutely most important thing we can all do in the fight against antimicrobial resistance is to try to prevent infection. If you prevent infection, you absolutely won't get a drug-resistant infection, right? So this is really simple, and I think COVID taught us all a lot about preventing infection. So this is when you're not well, it's staying at home, it's not passing it on to other people. It's good hand hygiene. So it's all fairly simple stuff, right? It's getting vaccinations when that's possible. So that you try and stay well and not get infections in the first place. We can avoid unnecessary antibiotics. We can think about whether we actually need an antibiotic, or we could discuss that with the GP rather than thinking that we should always be getting antibiotics when we have an infection. We should always take our antibiotics as they're prescribed and never take an antibiotic that's been prescribed for somebody else or prescribed for you previously. And talk about it, spread the word. So hopefully you will leave this evening having a better understanding of what antibiotic resistance is and why it matters. And it would be really great if you could talk about that with other people too. And with that, I'll finish, and I'm really happy to take any questions.
Right, so I can roam with the microphone.
I'm a vet.
Oh, hello.
And we when we're prescribing antibiotics and other drugs, it's usually milligrams per kilogram.
Yeah.
And yet, if my sister and I require treatment for the same thing, she weighs 65% of what I do. Am I being underdosed and she's being overdosed? Mm-hmm. Another point is that the veterinary profession has made big strides in reducing the amount of antibiotic use in animals. Yeah. And thirdly, sorry to go on, in many countries you can still buy them over the counter.
Absolutely. So let's take your points. I think you're absolutely right. I think your first point about how animals are dosed per kilogram makes total sense, right? And we don't have that in human medicine, and yeah, as you say, who's being underdosed, who's being overdosed? I think in that respect, vets have absolutely hit the nail on the head. You're absolutely right. In veterinary medicine, the strides have been huge, particularly in the UK. In UK agriculture, enormously, the amount of antibiotic being used is absolutely reducing. And then your third point was
about buying over the counter.
Buying over the counter. You're absolutely right. And so we saw variations in the rates of resistance around the world. You will also see really big variation in how antibiotics are sold and prescribed. So here, you can't buy antibiotics over the counter. You have to be prescribed them. But that's absolutely not the case in many places. It's very easy to think, though, that we should stop that happening, we shouldn't let that be the case, but actually there's a balance between access to drugs and people that need drugs being able to get them, and overuse of antibiotics and using too much. So you're right, and that's definitely a contributing factor to resistance in many parts of the world, but politically perhaps not a straightforward one to solve without cutting off access. So, yeah, really interesting questions. Thank you.
Hello.
Hello.
If your Superbug it's got one of these efflux pumps, how can you tell? Can you see it? Can you DNA it? And how many of the Superbugs are likely to be defeated by this, and have any been defeated so far?
Great question. So, in terms of bacteria, when we're talking about the gram-negative bacteria, which are things like E. coli, Salmonella, Campylobacter, Pseudomonas, things you might have heard of like that, they all have these pumps. The difference is that when bugs become resistant to antibiotics, one of the things they do is they make more pumps. So rather than just having one, we would have an army of people pumping out the water, and they would be pumping it out much more quickly. So have they been defeated by them? Not yet. So we can find molecules in the lab that inhibit these pumps really well. So really effective cable ties that stop the efflux pumps working. What nobody in the world has yet managed to do is find a cable tie that is also suitable to be given to humans as a drug. So while we can find molecules that do the thing, they don't do it yet in a way that's safe to give as an antibiotic, to give with an antibiotic as a drug. And that's what we're working on. What we're actually trying to do is think outside the box and think about different ways to inhibit them, so different ways to make cable ties. But, you know, it's a watch this space, and it's something that lots of people around the world are working on.
Thank you very much. One of the things about antibiotics, of course, is that we like prescribing them in round numbers, like 5 days, 7 days, or 10 days, which of course is entirely arbitrary. And I was going to ask you how much of a problem you think people taking not the full course is. Likewise, how do you really know what the full course should be?
Yeah, great question. So actually, the evidence on this isn't especially clear. In that you're right, they were relatively arbitrary numbers when that have been set out as what the course is, and there is now research looking at what the appropriate course is in terms of the best clinical outcome, but also the right amount to do as much as we can to prevent resistance. And the second part of your question was.
It's when people feel better.
Yes, right, and they stop taking them, because also some antibiotics can actually make you feel a bit lousy. Now, there's all sorts of reasons for that. Some of them are a bit nasty. Some of them will also kill off lots of those other 100 trillion bacteria which are in or on our bodies that we rely on, right? So yeah, sometimes antibiotics can make you feel lousy in a different way to whatever the reason was you were taking it. And if your sore throat gets better or your infected toe gets better, you might stop taking it. I've got to say that we recommend that you don't do that. Because actually that could contribute to antibiotic resistance, because you're more likely to leave behind your check shirt bacteria, right, in that scenario. Again, the evidence on this depends on the drug, it depends on the type of infection as to how big an impact that makes. But the advice would always be you're better to finish the course, partly because of resistance, but partly because you're more likely to make sure that you stay well.
Any more questions?
Yes. When antibiotics were first discovered, who decided which illnesses to try them on? And how long did it take before the general picture emerged on this?
That is a great question, and one I'm not totally sure I know the answer to. I think almost certainly what will have happened is that they will have tested in the lab which bacteria penicillin was able to kill. So that would have given a good idea about which ones might have been treatable. Do you know any more about that, Gavin? Sorry, I put you on the spot now.
It's a very good question. I think you're right, probably because you would have done sensitivity assays in the lab and then enrolled them.
And to be honest, probably trial and error, right?
Yes, I've got a rather strange interest. One of which is leprosy. Okay. Because I know about sulfonamides because first seaside holiday was on a liver colony,
Ah.
staying with the doctor who was trying these out for the first time, after which leprosy became treatable for the first time. So obviously these were available, or he would be able to test them out. He did some of the sort of first important clinical tests on it.
Yeah, you know, absolutely fascinating story around the leprosy colonies and being able to treat those. And I think with many of these things, particularly in the early days, would have been trial and error to find out what could have been treated. Because at the time, there probably was less understanding than we have now about things like, will the drug get to their site of infection? Will the drug be stable in the body for long enough? These things. So actually, the only way you would know that was to try. Yeah.
Thank you very much. Sorry to be here. I mean, in relation to your last answer, of course, we're all very interested in our microbiome nowadays, and how much research is being done into preserving the microbiome whilst killing what you really need to kill.
Yeah, great question. So our microbiome is the bugs that live inside our body and are a really important part of keeping us healthy. Okay, so our gut microbiome, for example, millions, billions of bacteria that live in our gut and we need for proper digestion and for really staying healthy. And actually, you're absolutely right that it's really important when we think about antimicrobial treatment that we start to think about preserving that. One of the things that the field is relatively keen on is the idea of narrow spectrum drugs rather than broad spectrum. Now, broad spectrum means that an antibiotic that would kill lots of different types of bacteria. Now, there might be certain circumstances where that's really important. If somebody's very ill with an infection and we don't know what's causing it, a broad spectrum antibiotic's a good idea. But a narrow spectrum that only kills certain types of bacteria could be a way to protect the microbiome, because actually it's much more targeted, it's going to kill the thing we want it to kill, and there won't be the same collateral damage to the microbiome. There's also loads of research, including lots of research at Birmingham actually, on the microbiome and the benefits it provides, what it's doing, and how it could be looked after. It's a really important area of research and something that we need to understand better.
I was just thinking, when people take antibiotics, how much of it is excreted and therefore gets into our sewage system, and then with all the effluents being put into the sea and rivers, how much sort of is that impinging on our ecosystem?
Yeah, great question. So I don't have numbers for you, but and it depends, because also it depends on the drug, right? So certain drugs are excreted at higher levels than others, but yes, absolutely. The more drug we take, the more obviously ends up in our body, but the more also ends up in the environment, and that's because part of it is excreted, absolutely, but also the production process of antibiotics can put them into the environment. Some of the uses in agriculture can put lots of drug into the environment. And this idea is something that there's been a lot of research on. It's something called the One Health sort of view on antimicrobial resistance, and it's the idea that, you know, what is the impact of antimicrobial resistance in the wider environment? It absolutely means that there are antibiotic molecules out there where we wouldn't really want them to be, and that means that more bacteria are coming into contact with antibiotics and evolving, learning, you know, to not be killed by them. So it's certainly an important thing to think about, and again, there's lots of research going on to understand what the impact is of the levels of antibiotics that we find in our environment.
Okay, one last question.
Just a quick one. So probiotics have become quite the big thing. Yeah. And what I've read is they probably aren't that useful. I don't know. But we've got antibiotics, but what about these probiotics?
Yeah, really great question. So that kind of follows on from the microbiome question we had before. So probiotics is a big industry, so you'll get lots of yogurts or yogurt drinks that tell you they're probiotics and they're going to look after your gut health. In many of those cases, the evidence isn't great that they are particularly protective, not going to do any harm. However, there's loads of research going on in how to make those better and how probiotics can be used in certain situations to really protect your microbiome. So there's absolutely really great science underpinning the idea of probiotics and particular uses of them. So again, there's research going on in with the microbiome at Birmingham where they're looking at different probiotics that can be used in, for example, neonates, so premature babies, and helping them establish a normal, healthy microbiome. So there's loads of work going on in that area, and it's very much a watch this space, but the evidence varies.
Right, I'm going to close the questions now because we've got lots of fantastic questions and we need to just relax. Just one quick segue though, that excellent question about host range and broad versus narrow range antibiotics that Jess answered very well. Very nice segue into the next lecture in this series by Professor Martha Clokey, because she's working on bacteriophage, and these are potentially the ultimately most selective antibiotics. They will kill a particular strain of a particular species. So that's a really interesting concept that we'll really learn about in the next session. So thanks for all of your questions. Can we thank Jess again for an excellent seminar?